So, all right, so yeah, we're looking at, we're going back to like plasma membranes, right? That's the cell membrane. And and we're talking about cell signaling, but you can't separate cell signaling and the plasma membrane. They're very much intertwined. And that's because, and they're not showing it here, because in your cell membrane, bless you, you have proteins. And those proteins will act like basically receptors for different signaling molecules. And I'll show you a bunch of pictures and get into the details of that. But that's why this is in chapter 5, where they're getting back to the plasma membrane. Think of an entire multicellular organism like you. You have a bunch of different organs that each have different jobs. Those organs are made of different tissues. Remember, a tissue is cells of the same type. So ultimately, you're made up of cells, and those cells have to find a way to communicate with each other. So the question is, how in the world do all of the cells that make you up, how in the world do they communicate with each other? And there isn't one sentence answer to that, but there's a bunch of different ways they do that. But they ultimately all involve the plasma membrane at one point or another. And unicellular organisms will also need to have communication. So unicellular organisms, if you have like a layer of bacteria, like a layer of bacteria, those individual bacteria cells also have a way of communicating and sensing with each other, kind of sensing where nutrients are at and things like that. All right. Let me get my cursor over here. So we talked about gap junctions and plasmodesmata a while back. And this picture is referring to something called direct contact. Direct contact. Direct contact is a type of local signaling. So local, it's like nearby. Direct contact, the cells are literally touching each other. So if we're looking at cells like ours, animal cells, we have like little tiny, basically these are protein channels, like a little protein channel, so what they're not showing, that can connect the cytoplasm of two cells. So think of a bunch of muscle fibers, or even some of our skin cells and stuff. We can have these gap junctions that allow cells to efficiently share messages between the two cytoplasms. They can kind of basically share cytoplasms with each other. And plants will have the exact same idea, but in plants it's the plasmodesmata. Ions are the main kinds of molecules that are shared between gap junctions and plasmodesmata. It's small molecules. That's why ions are being shared, because an ion is just an atom that's lost some electrons or gained some electrons. So small molecules can diffuse between those gap junctions. If it's a larger molecule like glucose that's made of like C6H12O6, there's a lot of atoms, those can't fit through the size of those protein channels in a gap junction. So it's going to be a small molecule. And that's mainly what they're saying here. So we call that direct contact. Adjacent means next to, so it's the cells next to each other. So there you go. All right, some other types of local signaling. So direct contact, what we went over, that's a type of local signaling. Two other ones that are worth noting, paracrine and synaptic signaling. So paracrine, anybody know what para means? Like that prefix para? Luke? Yeah, next to. I think it literally means next to. And then crin is referring to like, I believe the secreting of something or something like that. It's like you're sending something out. So you're sending something out, you're secreting something next to something else. So paracrine signaling, it's basically cells that will have these secretory vesicles. Where do those secretory vesicles come from? The Golgi, yeah. These came from the Golgi, right? There's my Golgi. They come from the Golgi, and then these green circles, those are your signaling molecules. So those are in the secretory vesicles. They fuse with the plasma membrane, and then they send those messages out, and they send them to the target cells. Focus in on the word target. Notice how they didn't just say cells. And I'll show you some more pictures of this up later, but like a target cell has to have a receptor that can receive that message. Think of like walkie-talkies. I don't know if you ever got a chance to play with walkie-talkies as a kid, or at least maybe you know how they work, right? That you have to set the receiver on the walkie-talkie to a certain radio frequency. Think of like receptors on cells a similar way. Cells have to have the right receptor for that signaling molecule. Otherwise, that signaling molecule, if it's a cell that's not a target cell, that signaling molecule would just like bounce onto its plasma membrane and just keep passing it by. It can't receive it. Be like you passing people in the hallway. All right, another kind of local signaling that I want you to know is synaptic signaling. This is how nerve cells communicate with each other. So it's a similar idea where like you have these secretory vesicles, but these red circles, this would represent a neurotransmitter. A neurotransmitter is just a word that's just referring to like the signaling molecules nerve cells will use to communicate with each other. So this is a chemical signal. It's an actual like molecule. And this, anybody know what this is called? Like this ending part of the nerve cell? Axon, yeah. And then what's the receiving part? Is another nerve cell. What's the receiving part of the nerve cell? Dendrite, good, okay. All right, anyways. So then this signaling molecule will go across, this whole picture is called the synapse. What is this specific space called? It's part of the synapse. You just raise your hand. Need it? Synaptic cleft. Yeah. Remember I told you earlier, like, if you want to like a intellectual diss is like you say somebody has a very large synaptic cleft. It takes them a while for like process information. It takes a while for those neurotransmitters to get to the next nerve cell, right? Anyways, now on what they're not showing is there would be protein receptors that would receive that neurotransmitter, and then this nerve cell receives it. If it receives enough of the neurotransmitter, it will then send a signal on to the next nerve cell. Anyways, those would all be part of what's called local signaling. So let me make sure I said everything I want to say from that slide. Oh, an example of some of those signaling molecules that like we talked about last class could be growth factors. So growth factors could be something that like cells in a tissue layer, like, oh, there's a cut here. So then the cells that are like right next to that cut, they could release growth factors to other cells telling each other, hey, we need to divide to heal up that wound. And again, local regulators, they're local. They don't travel long distances. Okay. And this is about the synaptic communication. All right, so then if there's local signaling, there must be then long distance signaling. So how does like my brain communicate with my toe, right? How do we communicate messages over a long distance? Well, the main way is what's called endocrine or hormonal signaling. So you've all heard of hormones, right? So hormones are the main way that we want to communicate long distances. And think about it. What is the system that connects your entire body, all the cells of your body to one another? It would be your circulatory system, your bloodstream, right? Your circulatory system, you may not have ever thought about it that way, but it reaches all of your cells. If it didn't, how would your cells ever have any oxygen to survive, right? So what that means is if I have like some of my like cells in my brain, my nerve cells in my brain, they want to communicate with like other organs throughout your body, they can release a chemical messenger, some sort of chemical molecule into your bloodstream, and that bloodstream will eventually travel to all of your cells. And if this hormone will go to all of the cells, but if it targets specifically that target cell, in other words, it's going to specifically target the cells that have that protein receptor that can receive that molecule. I'm going to say that again because I think it's an idea that you might gloss over. This hormone molecule travels to all of your cells, but only the cells that need to respond to it would respond to it. Think of like the puberty hormones, like testosterone and estrogen. Not all of your cells care about testosterone and estrogen, but like there are obvious cells, like cell types in your body that are very important in puberty, they are going to be the ones that have the receptors for that hormone, for that molecule. Okay. I think that's mostly everything here. So make sure we set everything on this. Hormones, it's a very broad word. There's a lot of different hormones in terms of size, shape, hydrophobic, hydrophilic, polar, non-polar. So I don't want you to think of like a hormone can just be like, oh, all hormones are kind of the same. There's actually a big difference in like the structure of them chemically. That's why there's like you ever heard of an endocrinologist? It's a doctor that like just specializes on like hormones. That's why there's like an entire field of medicine going through this. It's very complicated, like how it all works. Okay. All right, so we want to go over the three basics of cell signaling. So there's a lot of different kinds of cell signaling. We won't get into all the different mechanisms, but they all different cell signaling has a basic process. The first process is reception. There has to be a receptor. Each receptor is specific. That's not how you spell specific. So in reception, there is a specific receptor for a ligand. A ligand is a fancy way of saying a signaling molecule. Okay? So each receptor is specific for a ligand, like a key would be for a lock. Right? So again, back to that hormone example, hormones they travel to all your cells, but they only impact the cells that have the appropriate receptor. So if you have no reception, there is no transmission of the message. If you don't receive the message, how can you ever pass on that message, much less respond to that message? Okay. And then the second part is going to be, one more thing on reception. Once this signaling molecule binds, so when you get like this complex, you get a change in shape of that receptor protein. So how this protein, like what's the connection between reception and transduction, the passing on of that message, is a change in shape of the receptor. So you don't even know the details of it, but something about this signaling molecule binding to that protein receptor will cause a change in shape of the protein, kind of like when you put a lock into a key and you turn the key, it causes a change in all the complicated gears that make up that lock that will send that message forward. Okay? Now, transduction, anybody know what that word means? Or like what it's kind of referring to? Like the basic idea of it? Because you'll hear transduction or something like that referred to in a lot of different contexts. Did it too?
It's kind of like sending the signal through sound.
Yeah, it has to do with like transmission. Or like transporting. It's sending it along, like that message to the next person, to the next thing in the pathway. So there's transduction pathways. Transduction pathways can be very, very long. Like, if you're curious, you can just Google like signal transduction pathway, and you can see like an actual like research paper, and you'll see like so many different molecules that are in this transduction pathway. So many, that's like many different kind of little middleman communicating the signal from the cell membrane to your response, where your response usually is the turning on/off of genes. And I want you to appreciate why that's important, because what comes from genes? Like, why would that be important we turn on or off genes? Why is that, like, important? What are we doing with the genes? Awesome.
We're using them to, like, build the receptors or build like amino acids or
proteins, yeah. Genes, each gene codes for a specific protein. Proteins ultimately are like the workers in the cell. So don't lose sight of what we're doing, the bigger picture. You have a signaling molecule. The purpose of that signaling molecule is to make certain proteins or not make certain proteins. That's basically what we're trying to do here. We're trying to turn on or off certain genes to turn on or off certain different proteins. That's what we're doing with these signaling molecules. A little bit more on transduction, and really like next week when we do the rest of 5.6, we'll look at some of the examples. But just to introduce this to you, phosphorylation is phosphorylation is a very common theme in transduction. So phosphorylation, that makes you think of phosphates. We just had our cellular respiration test and photosynthesis test. Where did we talk about like phosphates and phosphorylation a lot in regards to? It had to do, but like what specific molecule in cellular respiration were we making that involved phosphates and phosphorylation? ATP, right? So, hey, we're basically in a signal transduction pathway. We're going to attach phosphates to different molecules, and that kind of passes that message along to the next molecule. The question is, where do the phosphates come from? ATP, right? That's why we made, not the only reason, but it's a large reason why we made the ATP. What we're not showing is a lot of where we're getting these phosphates that turn on and off these different protein molecules, they come from the ATP. So I hope you're kind of making that connection. All right. I think that's everything I want to say here. Yeah, all right. Don't worry about glycogen phosphorylase. In your textbook, they use like a epinephrine as an example. I won't go into the details of that, but if you're curious, if you read your book, that's what they're referring to there. Anyways, if you care, the guy who discovered this was Earl W. Sutherland. I kind of like that name, Earl. I might name my third kid that. Anyways, I don't have two kids. All right. 3 processes: reception, you detect the signal; transduction, you convert that signal to a response, where you got to communicate that signal to where you're ultimately trying to get a cellular response, which typically is the turning on or off of a certain gene. As I mentioned, when you're in reception, the signaling molecule is called a ligand, and receptors are specific, like a lock and a key. When a ligand binds, you get that change in the shape of that protein receptor. Yeah, there you go. And the location of these receptors are the plasma membrane. That's cell membrane. All right, so then the last thing I want to talk about are G protein coupled receptors and ligand-gated ion channels. Generally speaking, you're going to have 2 different kinds of signaling molecules. Those that can't cross the membrane and those that can. Okay? So water-soluble molecules can't cross the membrane. Why? Why would a water, so soluble means it can dissolve in. Why would something that can dissolve in water, a signaling molecule dissolves in water, not be able to get across the cell membrane? Think back to like the structure of the cell membrane. Yeah, go for it.
Part of the membrane cycle.
Yeah, that inner portion of the cell membrane. So like if I do my basic cell membrane, it's that lipid bilayer, and the tails of the lipid bilayer. This portion right here is hydrophobic, right? It has a fear of water. It is non-polar. Water is polar. Water can't cross the cell membrane. That is why you have to have these protein receptors, okay, to transmit that message. These protein receptors, they span the membrane. What would that mean about the structure of receptor proteins that allows them to span the plasma membrane? And we talked about it earlier in the year. What these proteins, we need to have what properties to be able to span both portions of the plasma membrane, the outside, through the middle, and then back into the inside. Good. Amphipathic. Amphipathic. That means they have hydrophobic or non-polar and hydrophilic or polar regions. Okay. Remember we talked about polar and non-polar amino acids. Amino acids make up the proteins. So these proteins, they have to go across the entire plasma membrane. They got to have a portion, the portion of it that sticks outside the plasma membrane. This portion would be hydrophilic, it's polar. The portion of the protein receptor that goes through the plasma membrane This would then, so this would be polar. This portion would be nonpolar. And then the portion that then sticks out into the cytoplasm, so this is the cytoplasm, the inside of the cell, this portion would then be polar. All right? Hopefully that's something of a review to you. But you gotta remember, like, we study units separately, but they all ultimately connect together. All right, so that's what's like can't cross the membrane. So then what kind of molecules can cross the membrane? What properties of a signaling molecule would be able to cross the plasma membrane? Thomas? Nonpolar or hydrophobic molecules. I'll just say NP for nonpolar, aka hydrophobic. Right? Molecules that are nonpolar or hydrophobic, or just small, like if they're small enough, they can, even if they're maybe slightly polar, they could still cross the plasma membrane. All right, anyways, I know it's kind of a lot going on there, but so this slide is showing you a G protein coupled receptor. And it sounds very intimidating, but if you tracked with me so far, it's kind of a straightforward idea. So here we have our protein. Notice how that protein like spans the entire membrane. A signaling molecule specifically binds to this protein. This protein, like that shape there, that only binds that signaling molecule. Once that signaling molecule binds, this GPCR, G protein coupled receptor, gets activated. Now notice the name, G protein. Well, this is the G protein right here, this blue thing. Coupled, what is a couple? How would you know somebody's a couple? What might be a sign people are walking the hallway that they are a couple? They're touching. Hopefully they're not just like making out, but like they're holding hands, right? Like they are coupled. There is physical contact. They're like weirdly close to each other. That would like beyond what would just be normal socially, right? And these two proteins are coupled to each other. Okay? And the receptor is coupled to the G protein. Anyways, this protein is activated when you have GTP bound. What does GTP remind you of? ATP. They're not the same thing, but the A and ATP, what was that? Adenine. So then, and what was adenine? What did adenine make up? DNA, right? The A, T, C, G. So what do you think the G in GTP stands for? Guanine. Yeah, good. So then this would be guanine triphosphate. All right, so once you have GTP bound, GTP, because it's an energy source, that acts like a light switch. That energy source turns on and activates that protein. And now, check this out. It's actually kind of wild. This G protein will just like straight up travel across the plasma membrane to another protein, specifically an enzyme protein. Now, what do enzymes do to reactions? They speed up. They catalyze reactions. Catalyze or speed up reactions. So once this G protein binds to your activated enzyme, you then get that cellular response. That cellular response can be a lot of different things, but the main kind of theme I want you to think about is the turning off/on of genes. You know, the making or not making of certain proteins. Okay, so this isn't the only mechanism. This isn't there's a lot of different G protein coupled receptors that do a lot of different things, but this is a very common theme you'll see in cell signaling. Okay, and many different species have G protein coupled receptors. Like evolutionarily, this is a very like kind of ancient system, they would say. And then if I want to turn off that signal, I need to remove my signaling molecule. You remove that signaling molecule, we'll stop doing that process. Or if I turn the GTP, like if GTP was the on switch, what would be the off switch to the G protein? What's the opposite? Not really like literally the opposite, but like how would you turn off the GTP? Think of cellular respiration, like the cycles. You had ATP, and then like what would you need to regenerate ATP? You need ADP. Sorry, I did get your hand up. Yeah, so you can turn off the GTP by breaking GTP into ADP and that inorganic phosphate. Anyways, so that's how you would turn off the G protein. All right, I hope you're still with me here, but yeah. So it should have said everything here. G proteins, they bind GTP. A lot of different kinds of G protein coupled receptor pathways, but they all these G proteins will all have a very similar function. What that means is if you change the structure of a G protein ever so slightly, that can really change its role or its function in the cell. All right, then the last kind of specific signaling pathway we're going to talk about are ligand-gated ion channels. So you see these a lot in nerve cells. And these are a lot more straightforward. These work by, you have your ligand, which remember is your signaling molecule. It binds to, and remember these are just proteins. It binds to a ligand-gated ion channel receptor, a protein. A protein that binds a ligand. Ligand-gated is a gate controlled by a ligand, meaning if that ligand binds, that will cause the gate to open once the ligand is bound. And then ion channel, so then this is a channel for what? A channel for ions. So as I said earlier, like the name literally says what it does. Ligand-gated ion channel. It's very helpful in that regard. Like you're not just like randomly memorizing people or something, you know? So there you go. Once this ligand binds, these ions come in the cell, and then some sort of cellular response happens. And I mentioned earlier, like genes turning on or off. That isn't, to be clear, that isn't the only cellular response that exists. Usually the cellular response for ligand-gated ion channels would be an action potential. Those of you maybe did like psychology or A&P, you may remember what an action potential is? They run over it. Thomas? Perfect. It's just textbook, right? The simplest way I can say an action potential is just like, it's like signaling, it's like a signal in a nerve cell. It's like, it's the minimum amount of charge that needs to like minimum amount of change in charge on the inside of a nerve cell that needs to happen for that nerve cell to send that signal to the next nerve cell. Not necessarily important for you in AP Bio, but just know like that's an example of a cellular response. Thank you. And just know like it isn't literally all just turning on or off of genes, but even though that is a very important process. Anyways, and then if I want to close the channel, I just remove the ligand. And that makes some sense. Okay? And again, the binding of that ligand causes a change in the shape of the protein. That's a detail I worry you forget about with like the reception of a signal. It causes a change in the shape of the protein. All right, that's most everything. Let me make sure I said everything here. Usually these ions are sodium or calcium. Sodium, calcium, potassium. Those are pretty important ions, and like especially like nerve signaling and like how muscles contract and stuff like that. All right, any questions? Okay.